Life Beyond PhD seminar series. As I always say, there is life beyond PhD. This is not the end. You can breathe once you get out of it, and Tyler will tell us a lot more about it today. So today's speaker is Tyler Korman. Tyler, it's great to have you back. Tyler is UCI alumni, and Tyler is currently serving as the director of our research and development at InDesign Technologies. It's a sort of focus on specialty and commodity chemicals using cell-free biocatalysis. You got your PhD here at Molecular Biology and Biochemistry Department. Then you end up doing postdoctoral fellowship from UCLA, James Boyce Lab. And that's where you basically built the core technology to start the company. Right? Thank you, Tyler, for coming, and it's all yours.
Okay, thanks for having me. Thank you all for coming as well. You know, it's funny, I was here not too long ago. I was in San Diego, and I stopped by about 3 months ago, and it's changed since I left in 2008. So I was here in Dr. Cheryl Tsai's lab, just across the way in Sci 1, right when that was built, from 2003 to 2008, where I studied structural biology of polyketide synthase enzymes. So, you know, it was one of those types of things where I came in, and I remember the first day of orientation, I said, you know, you see all the presentations from the professors, you know, looking to take people, and she had just started. She didn't have a lab. There were no people in it, and it was, these are polyketide synthase enzymes. They're involved in making drugs, you know, different antibiotics, anti-cancer things. I want to study the structures of them. I said, that's what I want to do, and I went into that lab, and from day one, just hit the ground running. I had come in with a master's already, so I had a little bit of training. I knew I wanted to work with enzymes and enzyme structure, and so structure-based drug design was exactly, she basically spoke right to me. So it gave me an excellent background over the 5 years that I was there in protein engineering, structure function analysis, published a number of papers. It was an overall great experience. After that, I went through the same thing that everybody else goes through. What do you do next? Kind of the lowest barrier is to do a postdoc. And so I had just gotten married. My wife wanted to move back to the LA area, so I looked around LA. And interviewed in a couple labs at UCLA, and took a slightly different approach than a lot of people will go into the biggest lab, the biggest name that they can find. I went into Dr. Bowie's lab, who at the time he was a membrane protein guy, is relatively big in that field, but this was a DOE institute that he was a part of, so the Department of Energy, and he was getting money through that and said, well, they want us to do more fuel-like projects. So how do membrane proteins relate to fuels? Not very much. So he's like, I have this idea. Why don't we engineer a stable lipase to be used in biodiesel production? And I said, that sounds cool. I had heard from Celia, who had been at UCLA as well, that his lab was great. And I said, okay. The only caveat was this project would need to be built from the ground up. So there was basically nobody had worked on it before. But I was used to that from UC Irvine, because as a grad student, there was nobody there as well. So I said, okay, let's do it. And it worked. But lipases for biodiesel is kind of hard. We published a couple papers. And then one day, in about 2013, Jim came in and he's like, you know, I have this other idea. Why don't, you know, metabolic engineering, people have tried to do, engineer cells to make things, right? And it all kind of works to a little bit, but why don't we just get rid of the cells? So what do you think of that? And I said, well, that's an interesting idea. Let's see what we can do. And I think both of us were just naive enough and, you know, crazy enough to say, okay, what if we just reconstituted whole metabolic pathways outside of a cell? So link something like glycolysis up to a pathway to build things like, you know, terpene production, and would it work? So we started doing that. We worked on that for a number of years, developed some cofactor regulation methods that work cell-free, engineered some new proteins along the way with altered specificities, and really built upon my experience with enzyme engineering and primary and secondary metabolism that I got while at UC Irvine, to kind of start this whole project where we were able to now turn glucose into some useful product, right? Along the lines, we're like, well, maybe we have something commercial here. This is a different idea. Metabolic engineering of cells is hard. We thought people should be interested in it, so we founded a company. That was maybe a little premature, so we didn't have a lot of stuff fleshed out. That was in 2015. It's now 2020. But what it did is it started us thinking in a different way about the research that we were doing, right? How do you take fundamental science and actually apply it to something that people want? And what are the things that should go into a research project, and what you're making, how you're designing certain things, to be something that people will actually pay for or want to invest in, right? And so natural products were kind of, we were lucky we got a Department of Energy grant to kind of help build this out. So it's nice to have funded projects that allow you to do that. And so we founded Invizyne, and it wasn't until kind of the end of 2018 when we engineered a prenyl transferase that does the last step in cannabinoid biosynthesis that everything kind of fell together. Because now we had a platform that works, right? That's novel, but we now had the application as well. So we were able to show that you could apply our platform and all these design principles to actually making a compound that people wanted, right? And so we opened doors very end of September, so October 1, I'll say, 2019. So I've only been there for 3 months. So this has been a long process since 2015. But we have funding. We also have now a number of grants as well to continue to build and do this. So my day-to-day, I direct research to apply the platform, manage research and grants and people. So maybe just briefly, if you want to hear about the science behind it. So we really want to just simplify biocatalysis. That's the whole premise. So like a bacterial cell, you can make genome edits, you can put plasmids in, turn a substrate into some product using that pathway, right? But this all functions in the context of the background of cellular metabolism. It's hard. So we said, what happens if you just get rid of the cells? Can you do this whole thing cell-free? So the whole idea here is that you can use sugar now, break it down into building blocks and high-energy cofactors that you can then use to build some interesting molecule that is some kind of useful compound. We want to do this all in one pot. And the most important thing, so sugar's cheap, enzymes could be cheap, but cofactors are really expensive, ATP, NADH, and ADPH. So you really need to be able to recycle those efficiently in order to have now our goal of an in vitro bioproduction platform that functions continuously for long periods of time, right? So that's the goal and the ideal. Most people have set up systems where you're stoichiometrically balanced, so the amount of energy you use is the same that you make is the same that you use. So our innovations kind of remove the bottlenecks associated with doing that. And at the same time, they're autoregulatory, right? So what that means is, again, if your breakdown phase makes NATP and you have to make your build phase use that NATP. If there's any spontaneous hydrolysis or oxidation, the whole thing shuts down. So we developed a number of schemes, which we've coined. There's papers that describe this. It's called molecular purge valve for reducing equivalents, molecular rheostat that manages ATP. They're all just drop in, and they function within the system, so there's no sacrificial substrates or anything. It's just sugar to product, and you recycle the cofactors. It allows you to build up an excess, actually, of power to then drive these other reactions. So we have a number of papers where this is all described with a couple follow-ups in preparation. We've applied it to a wide variety of different chemicals. So we've made biofuels, so isobutanol at well beyond the cell toxicity limit. We've made flavors and fragrances like terpenes. So limonene is the monoterpene from like lemons that kind of gives us this lemon smell. So we've done that at quite a high level as well. And perhaps the most interesting to many people, we've also made now cannabinoids at, we're actually well past the multiple gram per liter scale now as well. All cell-free, right? So all just dumping in enzymes, low-cost substrates, and a little bit of cofactor, and having it just produce the product that we want. We also have a number of patents granted and provisional. So this was all done at UCLA, right? So always keep in mind, like the stuff that you're doing, if it has a use, you should be telling the university, right, that it's out there and trying to patent it because people might be interested, or at the same time you might be interested in your own startup as well, right? So a lot of this doesn't get done unless you kind of have control over that background IP. So kind of our motivations moving forward. So we have this cell-free biocatalysis platform we think simplifies the whole prospect of engineering and building these processes to actually convert low-cost substrates into products. So the basic premise is, can we use isolated multi-step enzyme pathways to do what cells do, only better, right? So this is kind of a small but significant change in the way even enzyme biocatalysis is done. So there are some companies that will do one-off transformations, like reductions and things like that for pharmaceutical biosynthesis, but it's just now changing to do actually multi-step enzyme pathways all at once. Can we apply this approach now to a wide range of targets, faster with higher titer and yield, right? So really speed up design, build, test cycles, and always being inspired by the need for constant development via enzyme and process engineering, right? So we always fall back on our ability to work with enzymes, engineer enzymes, and to get them to do what we want them to do. So again, we were founded in 2015 to commercialize this approach. At the time that we founded it, we only had the regulatory elements, and we were searching for good commercial targets. In late 2017, we developed this specific prenatal transferase. And for my part of the story, this goes back to having a conviction in what you're doing. Because that initial idea, I brought it to my boss at the time, and he's like, it's not going to work, right? And I'm like, I think it's going to work. He's like, no. So we wanted to computationally design this protein to bind a substrate that it didn't want to bind. He's like, you're just going to design an inhibitor, right? It's just going to a protein that's super inhibited. I'm like, whatever. With how cheap it is now to synthesize enzymes, I'm like, let's just design it and try it. Like, it'll take 2 weeks. And it literally happened within about 6 to 8 weeks. We had data, and it worked. And we were like, okay, let's go forward. It enabled us to raise some private money in early 2019. We opened doors in Monrovia in the fall, like I said. So Monrovia is just east of Pasadena by Caltech. And along those lines, we've also now, we have government money. We have an SBIR, which is a small grant. We have a larger DOE BETO grant to develop the biofuel part of this as well that were just issued. And hopefully more to come. So we have some other grants out there as well. Currently, this is the team. So the founders are myself, Paul, and Dr. Jim Bowie at UCLA. We've hired one scientist, also from UCLA, John Billingsley. And we're looking for good people to join our team, protein engineers, process engineers, molecular biologists, technicians, so really people at all levels to help us build out this platform for both the BETO grant and the other things that we're doing. There's my email address. If you want to write down and reach out later, that's fine as well. And that's all I've got. So hopefully it wasn't too fast. And if you guys have any questions, I am.
Yes, go ahead.
Can you talk a little bit about that transition from working for UCLA and transitioning to like your own company, in particular like the patents and how that when did those happen and that kind of like that process?
Sure. So it wasn't necessarily a smooth transition. Like it wasn't like I'm doing this at UCLA and then boom, I'm here at the company. So there was some overlap. Some of those early patents were as early as 2014, right? And so that whole process of patenting the work that you're doing happens at the PI level, right? First, like you have this research, you've written up the way that we did it. You wrote up a paper, you submit it to the Office of Intellectual Property first. They submit a provisional patent, and then you submit the paper, right? So that you can make sure that you keep that priority, right? So you didn't publicly disclose any of that research before you patent. Sometimes, if you think you have a big enough idea, you might want to push your boss to say, let's patent this earlier, right? Before you've even written the paper, just to get that early priority date. So along those lines, like once it gets filed, there's a long period of waiting, like a year, right? And then the patent office and the lawyers that the university has will get into contact with you for feedback and things like that, and then you kind of go back and forth to get the patents squared away. Once that happens, you might not realize, but now you have to actually license your own technology back from the university, right? And you actually, if it's UC Irvine, you can't do that yourself. So you need a third party who's not employed by the university to do that negotiation for you. It could be a lawyer, it could be a CEO, a partner, whatever. And so that was one of the things that we struggled with because I was a founder and I was a UCLA employee. So I couldn't be involved in the negotiation of the license, and that's like a whole separate thing. But once you've got that, then you can go out and open doors and start your company. So,
Yeah.
How helpful or harmful was UCLA to the process?
of patenting,
Yeah.
um,
Starting the company?
so UCLA had a startup in the box program where they would put you in contact with a lawyer for free, and then they would do pro bono to help you incorporate, so that was nice. But there's really no guidance on what direction you should take or market research or how does your product fit. You have to do a lot of that yourself. And so they were helpful to a point, and there's a lot of technology that comes through, a lot of different technologies, right, that comes through these offices. And so, and each university is going to be different. So some are going to be really helpful and really involved, and depends on the technology that you're developing too. Like Berkeley is fantastic at a lot of these synthetic biology applications. UCLA, if we had a medical device, they would have been fantastic, but they didn't really know where we fit, right? So we had to do kind of a lot of that on our own.
Yeah. What kind of skills are you looking for in hiring people?
So, I mean, definitely a team player, right? First and foremost, like fitting within, likes to work with others and ability to share ideas. But our current, like most pressing needs, protein engineering. So somebody that's worked with proteins, expression, assay, characterization, that kind of thing. And then we could also really use a process engineer. So somebody who has done like separations or has experience, maybe more of a chemical engineering background or chemistry, those types of things. So that's, at the scientist level, that's kind of what we're looking for. At the technician level, again, somebody who's done the molecular biology, cloning, protein expression, protein purification, that kind of thing.
Tyler, I have a quick question. Did I write it? Yeah, the main difference between working in a startup versus large companies, and why startup?
I think startups are all going to be as, different as can be, right? Different as people are. It's definitely a different environment, even compared to, like a lab that's putting out a lot of work as well, where you're tied to deadlines and things like that. A lot of times, there's less people at a startup, right? So there's 3 of us. So we have to do things like clean all of the dishes, do all of the ordering, right? Figure out like where the trash goes, right? So some things that you wouldn't necessarily have thought about, but you kind of got to do. That being said, like well-funded startups are sometimes great because now you have like money that you should be able to go and say, hey, let's do this and let's go fast, right? So the speed at which things go and are required to go at a startup is very fast. With a large-scale company, it depends on what you're talking about with a large-scale company. So there's different flavors of those as well, right? So you have like, if you guys are familiar with maybe like a Genomatica, like a medium-sized, I guess, they're not technically a startup anymore, but they probably have anywhere from 25 to 100 people, right? They are still looking for new ideas and new things to do, and they work relatively fast. Somebody like BASF or DuPont, right? They're going to go very slow, right? Almost as slow as like an academic institution or the government, right? So it's just different. I'd say definitely a postdoc is not necessary to get hired in an industry research job. Again, sometimes it falls. I will admit, the first place that we looked, or that we are looking, are people that we know, right? So that ability to network in, either at a conference or people that are in other labs that are moving elsewhere, like keeping those connections active, right? Even if it's just an email or 2 once or twice a year, right? Sometimes can help, especially if they're doing and working in an industry or an area that you think is really cool, right? That you could see yourself. It might seem self-serving, but it doesn't have to be. It can just be like, hey, like, I think what you're doing is really cool, and like, if you're like at the same time doing good stuff on yourself, right? I think that just goes even further because now you just know everybody, and that's usually the first place that you're going to look because these people already know you. They're comfortable with who you are, the way that you work, things like that. when I decided to do a postdoc, I never thought I was going to stay in academia. That's the caveat. I never thought I was going to be a professor. I loved being a project scientist. I thought that job was fantastic. And if you can find it, and if you can find a PI that's really good to work with and that has money that's funded, sure, you're not going to make the most money, right? Salaries in industry are typically higher, but it's a great like work-life balance, right? You have a project that you're working on, it's yours. You're at a level in your career like where you can actually do stuff on your own and have your ideas, and like that was great. It just happened that this was a fantastic opportunity for me, that the stuff that I had been doing was commercial and my boss wanted to be involved and to move it off campus, and the stuff that we were doing stopped being academic and it started being more, how do we scale this? That's not too interesting, right? It's not as interesting as how do we get these things to work, right? And so that's kind of, we moved over there, and like that's been fantastic. And so that is really what inspired us to take the startup route. Early on, my bot Jim had asked, would you ever be interested in starting a company? So I always knew that that was in his mind. And when of course, what I said was like, sure, that sounds like a good idea. I never knew how much work it was going to be, right? And like, just as with anything, how many mistakes you're going to make, right? So what really lit the flame under us to get us moving forward, like in all earnest and seriousness, was I had gone to a conference in 2016, maybe, like a Gordon conference, right? And while I was there, I sat down next to a guy that was from a company. I was like, I've been doing this stuff. And we had already incorporated our company. I said, we've been doing this stuff. It's really cool. He's like, oh, well, we might be interested. And I'm like, you should talk to UCLA to maybe get a license. I thought that would be great, right? It'd be good for everybody. So he contacts the Office of Intellectual Property. And the university's goal, at least UCLA, is to recoup their costs. So they will license, like, they will get that. They will take that money. But that's bad. That would have been bad for us because now this other company would have had access to our IP. And so we actually had to work a ton to insert ourselves back into the middle of those two. And now UCLA knew that they had a fish on the hook, right? So we had to convince them, no, you should license it back to us. We were the developers, and let us do what we want to do with it, and then we'll make the decisions about licensing it back out. But in doing that, they're like, well, we will do this for you, but you have to actually work to raise money, show that you're serious, right, to move this technology forward. You can't just sit on it. So that's kind of the startup mistakes that you may or may not make. The main difference between an academic and industry research job in terms of lifestyle, my life hasn't changed from a research standpoint, actually. Like, the stuff I was doing at UCLA is pretty much the same as the stuff that I'm doing. I was in a fast-paced lab at UCLA. The people that I'm working with, one came from JBay up in the Bay Area, one came from another lab at UCLA, and they continue to work hard. So I would say that hasn't changed. I make a little bit more money now, which is nice. If you start my career, would you do anything different? I would say no. I might be different than a lot of people. I've always gone where I knew I would be happy, right? So I always wanted to like work on the stuff that I wanted to work on, and with like in a good situation where there was a PI that had good funding and was not like, over demanding. And like I really landed in a place where like I kind of had the freedom to do what I wanted within some bounds, right? But at the same time, I always enjoyed what I was doing, and that's always been the most important. The skills that you need to develop, you need to be able to talk to people. You need to be able to present your research and what you're doing in simple terms, right? I think that's not necessarily something that a lot of people think about. To a certain extent, you I can no longer rely on a PI to be making decisions about the research direction. So it's like I look at the wall, I'm like, is this a good idea? I mean, we have other people involved, right? But you have to have a confidence that what you're doing is right and that it will work, right? And so a lot of that comes with experience, and some of it comes with trusting the people that you're with as well, that you're working with, right? So that's just as important in the startup, because there's so few people, you really have to trust each other that you're doing the best that you can and that everybody has good ideas and the things that you do are towards a goal, right? Whereas in academia, as a research scientist, you have a little bit more freedom to kind of do stuff that is kind of off the hook, that like may or may not work. And if it takes you an extra 2 or 3 months and you have that time, like sure, that's okay. The biggest mistake we made, I mean, you always run into issues with, and this was not I haven't been there long enough at the company to have big mistakes or regrets yet. I mean, at UCLA, sometimes you can bet on the wrong people sometimes, but that wasn't necessarily my decision in the end. The biggest kind of mistake was actually the thing that got us moving the fastest, which was saying that we would license this technology to another company and then having to license it back to ourselves and insert ourselves. We could have done that much smoother, I'd say. So my working hours have changed, kind of. So like I said, like I've always been interested in doing things that make me happy. Part of that is making my family happy as well, because I have 2 kids, I have a wife. And so I commuted from Pasadena to UCLA for 10 years. So I would leave at 6 in the morning on a van pool. I'd go out there, and then I'd leave UCLA at 4:30 in the afternoon and be home at 6. So those were like 12-hour days for 10 years. One, I had a boss that was okay with me doing that, which was fantastic. And now the startup, I don't even have to get on the freeway. So in some ways, like, it's fantastic. I definitely work more hours now, like hands-on in the lab. But I'd say like 8 to 10 hours is pretty normal. Like I can come in on the weekends a little bit more now. It gives me more flexibility to kind of stop in and do stuff, so that's nice. But some of my coworkers live further away and they make it work as well. Yeah.
What do you see through the vision of your future of the company? I mean, you mentioned these products, cannabinoids, seems very sexy kind of thing, but do you see, I mean, it seemed maybe that it was a bit fortuitous that you were able to get this enzyme to work to do that, right?
Yeah, so there was a conscious decision to actually modify the enzyme we did to do that specific thing. Like that was the specific target that we were going after. And part of that was to get a nice research publication, right, high profile. Part of it was also we knew that, yes, it would be an interesting molecule commercially. The thing with cannabinoids is they're one type and class of compound. But for the company generally, since it's a platform, we can use that same technology. So it's based off of core technology. And we can use this technology to do the same modification for a wide variety of molecules. And we can also use the same core technology to do other types of modifications as well, right? So there's a lot of other types of chemicals that are difficult to produce or impossible to produce in microbes, or that are just so low natural abundance in plants that you can't extract them efficiently.
So you feel like the approach is pretty robust in making these enzymes and getting them
Yes.
to work, even
Yes.
though despite what your advisor mentioned, that it might be difficult by just making an inhibitor.
Yeah, so I think there's a couple things that we have going for us. Part of it is natural diversity, right? And the other thing is that with kind of the advent of low-cost sequencing and the ability to synthesize DNA really cheaply, you can do a lot of things really quickly just at the tips of your fingertips. So if you have an enzyme that doesn't work, you can very easily go and find 10 other variants that might get you closer and use that as a starting point. And you can do that on the time scale of weeks instead of months or years, right? And because you're not having to engineer the whole organism now around that metabolic pathway to make a single product, right? Now you can make different products simply by switching out a single enzyme. that whole process is really fast. So you can use the same upstream, change out a single enzyme, and now make a completely different product. And enzyme expression, characterization, purification is pretty robust. And again, if you have an enzyme that works, but maybe doesn't express very well, it's very easy to either design around or just screen natural diversity to find ones that will express or behave better, right? So there's a lot of reasons why you might think of just using enzymes themselves as opposed to the whole chassis of a engineered organism. You don't have to worry about ribosome or promoter strength or any kind of things like that, right? It's just, does the protein express? Is it active? So limiting to choose your career to choose a postdoc lab, I mean, maybe, but at the same time, I just wanted to do interesting research. There are so many labs around the country and even in the state of California that do cool stuff. I knew I wanted to work with enzymes and do enzyme engineering and things like that. So I don't think it was limiting. To a certain extent, I think it was fortuitous that I picked the lab based on, to a certain extent, the personality of the PI, right? That freedom to try ideas and think outside the box, and Jim's like okayness with once we had kind of felt each other out for the first year or two, right? Like he knew that I could do stuff. So it was like, well, now you start trusting in the decision. It's like the freedom to put those ideas to practice, right? And so I think it was fantastic. And I mean, it was one of the best decisions I've made, really. What other fields of science do you think are very exciting? Besides this kind of trend where you're replacing metabolic engineering and other forms of chemical catalysis with enzymes, things like 3D printing. There's some gene therapy stuff that's really, really cool that's out there. I do a lot of work, actually, things that I don't think a lot of PhDs and postdocs think about. Like even before, it was after we incorporated. One of the things that I would do is I'd spend a couple hours a week just looking at what other companies were in the field that were doing the similar things that I was doing. Which was, I'd go to a couple conferences and people were like, well, what company do you work for? I'm like, no, I'm a project scientist. And it was just like you could speak to what they were doing and what they might be interested in, like outside of your own kind of research. So, I mean, other fields of science, I know a lot about the other companies in my field, just because I'm interested in what they're doing, and there's some people doing some cool stuff. But I don't have a lot of time to think of like a lot of the other fields of science. Sorry, yeah, it's not.
What's the biggest challenge your company is facing right now?
So there now start to be issues with, it's no longer just about whether or not the science works, it's how much do things cost, right? Like, what is the cost of an input? Can I get that cost lower? How low does it need to be in order to make this commercially viable, right? So that's probably the major thing that we talk about on a day-to-day basis and that we are trying to address. Can you get it synthetically? Can you make it yourself? Can you get it from some third party some other way, right? Because it all comes down to the bottom line, like how can you get it cheaper? How can you make it faster?
Just to actually piggyback on the same question, when you were describing what's pricey, the ATP, in my lab I can get it for $10 a small bottle. What are you talking, what's the scale of price?
So think about it on a kilogram scale, right? So ATP is actually pretty cheap. So you can actually probably buy from somewhere besides Alibaba ATP for somewhere around $300 a kilogram. So it's not one of the major costs, although it does, if you're trying to make a commodity chemical for, say, $10 a kilogram, the prices don't match up, right? But if you're only adding a gram, so now it's 30 cents that you're adding in as long as you're recycling it, now maybe that process can work, right? So we think of everything now in terms of dollars per kilogram, right? So our cost of inputs are, and it depends on how well they're being used, what the stoichiometry is, and things like that. So, so yeah, that's kind of, I think, what a lot of people on the commercial side think of in terms of cost is dollars per kilogram.
What is like the most expensive part of your process?
The input's one of the substrates. Yeah. Well, it depends on if you're talking about now costs or future projected costs. Because enzymes and enzyme production is an issue that everybody in the space of enzymes has to deal with. Some enzymes are really cheap. So lipases, you put them in your detergent, right? They're pennies per kilogram. Specialty enzymes that are used as like biologics, like antibodies, are millions of dollars per kilogram or more, right? So there's a wide range. BASF and some of these other big companies, Novozymes, they can produce, I think, a wide variety of enzymes for anywhere from $10 to probably $500 a kilogram. And so that's kind of where we're working. And so with those kinds of considerations, the inputs are still, some of the substrates we still need to give it.
What's your opinion on patenting versus trade secrets? It's my understanding that someone can take your method now that it's public and you have really hard time knowing peer review.
Sure. And that is also, so we have a couple other things that we're developing concurrently at the company, right? And we have to make decisions on whether we patent it, whether it is patentable, or whether to just keep it as a trade secret. How likely it is that somebody else is going to develop that technology on their own. And so that is a consideration, and it's usually done with everybody involved. So all the members, the higher level, the CEO, the directors of research, along with the patent lawyers on the UCLA side and our own as well. So that's an active exercise that I think all companies go through.
I had a question about testing the market before you developed the company. How did you go about that?
So NSF has this, if you apply for SBIRs or if you want to be an entrepreneur, you have an idea, they have this I-Core program. We did not go through that. But the steps that we went through were pretty similar to what that is. So what that is the program, they have a smaller one initially, and then the I-Core one is a more intense, where you basically just cold call companies that might be interested, you think your idea might fit. And that's effectively what we did, is we just talked to people that we thought were maybe taking a metabolic engineering approach, making a certain product, and we're like, hey, what if we could do this? Or what if we can make this, would you be interested? Right? And so the more people you talk to, you start to kind of hone that idea and say, oh, don't really seem to be interested in this aspect, but this aspect they really seem to take on. And so then you kind of move into that space and maybe focus a little bit more on that. And so that's more or less, to a certain extent, cannabinoids were a little bit, the bar was pretty low. But moving forward and beyond that, right now is related to those types of discussions, right? So the more people that you can talk to without giving your idea away, right? Which is also part of the game, right? So nobody's using our products yet because we're not selling anything. But you can think of clients, and it depends on how you view your technology. And this is something that you probably need to think about if you're going to start a company. Are you going to make a product that you're going to sell? Or are you developing technology that you're going to license out? Right? So those are two very different business models. And that's another thing that I never thought I'd have to think about as a PhD student and then as a postdoc, is coming up with a business model, right? And you can find them online, and some of them are good and some of them are bad. And it's like, how do you actually apply this to make money? So I'd say our potential clients we're pretty agnostic at this point. Like, we have targets that we could make and we think we might be able to sell. But at the same time, we have other conversations with larger companies that might want to license the technology to maybe co-develop some product as well. So,
what resources do you use to get private funding for your company?
So we were actually, the private investors that we ended up going with. We actually met at a conference, and they approached us, and it just ended up kind of being in line. But one of the exercises that we were doing as we were going through this licensing the technology away from UCLA and trying to commercialize it was talking to people who knew about us and had reached out, or either had reached out or knew about us through publications, right? Because that's their first interaction with your research, usually in your science. And then saying, hey, do you know investors, or are there people that we should talk to? And kind of going down that chain. And eventually, most investors are willing to talk, right? And so if you can find a connection of a connection that, they will always pick up the phone and hear your idea. And a lot of times, you just have to do that a lot of times. There's some conferences that you could go to, so that was another resource that we tried. There's some conferences that are geared towards, not necessarily startups, but just business and science in general. So the Bio organization has an international conference where they actually have, you can meet with, set meetings with investors, potential investors, and things like that. UCLA did have some resources as well that we tried. So their technology development group, which is where all the IP goes, also has a vested interest in trying to move this forward. And so they often have a network of investors and things that they can put you in contact with. So I would assume that UC Irvine has something similar.
I had a question about your training. So when you were in Cheryl Tsai's lab, you were interested in enzyme engineering and went to go do a postdoc. What was that? What was your intention for that training?
I don't know that I necessarily had an end goal in mind. I wanted to continue to do protein engineering, and I wanted to focus on, because while on the face of it, a lot of the research in Cheryl's lab can be very applied. At the time, that was kind of in the far distance, right? It was all basically, what does the protein look like, and how does it do the reaction that it does? So very fundamental, right? And so when I was looking for a postdoc, I wanted a project that had more of an application, right? I didn't really want to do just the basic research anymore. And that's why I chose to go with Jim's lab, because it was an applied project, how basic or not, right? It was, you were going to develop a lipase to actually do something, as opposed to figuring out how an enzyme works, it does its reaction, right? So that's what I had in mind, and it was something that I could see myself doing. So one of the other PIs, who was a much bigger name at UCLA, who I also could have gone and worked for, I couldn't actually see myself doing the same, because it was solving basically similar structures over and over again. And I was like, I've solved some structures. I can't imagine, just doing the same thing for 2 years or 3 years, So it was a conscious choice on my part to do something that was applied, and that I could just be like, this is mine, and I want to do it, and I think it's cool.
And I just had a follow-up question on going into your postdoc. What was the timeline? Did you meet Jim before to make that connection, and then ask?
Yeah, so I don't know if my trajectory was typical. So I had started at UCSD. I got a master's there, so I had a lot of the skills, protein purification, cloning, that kind of stuff. So when I came to grad school, there wasn't a lot of training. The initial first 2 years, wasn't getting up to speed. It was I could go. And so within about 3.5, 4 years, I had enough stuff to say, okay, I'm going to graduate. And Cheryl at the time, I think, needed people to graduate because she was early faculty, right? So that's kind of part of this. And so I knew I was going to graduate. I had a date maybe set 6 months in advance. And within that time frame, I started, the first place I started was talking to other PIs that were on my committee or that I knew that were in the building and saying, hey, who would be good to work for? Do you know anybody that does this type of science that's in the LA area? That was one of my criteria. And then I just reached out. I cold called. I didn't cold call. So I made up a packet that had highlights of my research plus the publications, and I emailed it and then sent hard copies as well. And it was probably only 5 labs that I sent it out to. And I mean, I at least had people call me back. So, you know, because you never know. I mean, people have different states of funding and, you know, it ebbs and flows. But, yeah. I had heard good things. So I pre-screened also the labs that I wanted to go to. Like I wanted to work for somebody that had money and that was established. I mean, these are considerations, right? Like it's hard to work for, you know, PIs that maybe are not as well funded or haven't been, right? So.
And just a question bouncing off that, when you went into your postdoc, did you expect to be in a startup afterwards? Or
No.
did you like have another trajectory in mind?
I didn't actually. So I knew I wanted to stay in LA. There's not a lot of options if you want to stay in LA. It just so happened that the lab I was in at UCLA, I got along really well with my PI, and he really liked me, so I was able to continue on. And I really liked being a project scientist. And I probably would still be a project scientist at UCLA right now if this other opportunity hadn't come along. And so I didn't necessarily have a set goal. I wanted to do this, I wanted to do that, I wanted to start a company. It's kind of, you know, this is what happened, and it's good. It's good for me. So yeah. I'm a biomedical engineer, can be your product pipeline. I don't know, we can talk. I guess it depends on what type of biomedical engineer you're doing, but yeah.
What's the biggest downside of your job now?
You know, I have to do a lot of administrative stuff because I'm, I guess, the most senior, one of the more senior people besides the CEO, and there's not very many of us. So, you know, at universities, they have a whole departments that manage grants. At startups, you don't. So managing a grant is not necessarily the most fun thing. So that takes an inordinate amount of time, especially when they're like milestone-based grants, where you actually have to do what you said you're going to do.
Did you have to take any financial planning or
You'd think, right?
financial training at all?
So no, but I would say writing, when we first applied for the ARPA grant when I was at UCLA way back when, like 2012, 2013, I was part of that process, right? So Jim would write the grant and then I would edit it, things like that. And as the years went, we'd write more and more grants together. And part of that exercise became writing actually the grant. And then also, along with writing a grant, you have to come up with a budget, right? And so part of that is looking at the budgets that have been prepared before, right? And cutting and pasting as much as you can and filling in. And the more you do it, the more you kind of figure out where to put things and like what an indirect cost is. And like, I still don't quite know, like it's, I do know, but it's not fun having to learn it, but you do have to learn those kinds of things. And it's important also though, with a startup to have some amount of financial support. So I'm not expected actually to know all the rules for accounting. We have somebody else that does that, right? And so that's, you should have other people that can do the things that they are trained to do, right? I can write a grant, I can come up with a budget, you can send the budget, like happens here at UCI, happens at UCLA. Those professors will come up with a budget and they send it to somebody to get approval, right? And then it comes back, this needs to be changed, this rate is wrong, right? So you still need those checks and balances to make sure that when you do submit it, everything lines up, right? How much more did I have to learn to start my own company? I had to learn everything, right? I mean, I had not done it before. So yes, things like, well, the patent stuff was a process because you submit the invention report and then the university kind of walks you through the steps, right? So I kind of know that steps now, but commercializing the technology, we had to kind of learn that on the fly. Like, who do you talk to? What do you do? Hanging out with money folks? Sure, like you have to be able to talk to people that might not have a science background and to explain why the science that you're doing is interesting, could make some money, is cool, is different than what other people are doing. So it really takes, having to think about things in maybe a little bit different way. It's not all about the science sometimes. It's more about what you can sell.
It's sort of like the roadmap that for your company that you see it going. So you have some funding secured. You're going to keep developing, what that funding was saying. Is it sort of like a cyclic process where you get results, you apply for more funding, or you change trajectories if things don't work? It's like in academia, it's like, okay, your hypothesis is incorrect, you find something new. But in this case, if this technology is the output, then...
Sure. I mean, there's always the pivot, right? That's what they call it in industry. So like, company will go down and develop a certain technology or product, and they're like, oh, nobody's interested in this. Or maybe the market can't support it. So now you have to pivot and go do something else. So one of the things that we're trying to do, we've identified some markets, right? But at the same time, we also want to constantly innovate and develop other potential avenues that we can go into. So it makes pivoting that much easier. And because we have a platform technology, I think we can do that, right? So, it is kind of cyclic. With the government grants, you apply for what you think you can get funded. There's smaller amounts of money. On the private side, sure, it's about hitting milestones. Then you go out and you raise another based on interesting results, right? So, I mean, in some ways it's easier because you just have to sell that there's the promise. You've shown enough of a growth in your hypothesis was correct. Now it's just an issue of if I solve these other problems to get it to that commercial scale, I just need this much more money to do it, right? But hopefully you've pushed the needle so far that now your company is worth more. So you raise more money and that kind of thing. So I would say in startups, that's probably not true, prior industry experience. I think it would help because you have an idea of what you're getting yourself into. But I don't know that a lot of people have worked for startups that are getting their PhDs. Actually, one of my co-founders did. He worked for a startup before he went and got his PhD. But I don't know that it necessarily helped him because we were learning on the fly anyways. I mean, I know at UCLA, I don't know what's offered here at UC Irvine, but at UCLA they had one of the programs, one of the fellowships actually required you to do a 3 or 6 months internship at a company. If you know people that work at a company that are in the area or something, I know the business of science and the pharmaceutical masters or whatever that program is, I know they have a similar idea because I had some friends in that when I was here, where you actually go and you work for a company like either for no pay or very low pay for some number of months. Like that's one way. Yeah, so internships, yeah. It's really hard as a PhD student or a postdoc because you don't necessarily have control of making collaborations or partnerships, right? But if your PI does have that or is open to that, that's also sometimes an avenue that if you know somebody that has some technology that might fit with what you're doing, you can be like, oh, like my friend works here, maybe we should talk to him, maybe there's something there. So, yeah.
Any more questions?